We've all seen DNA testing portrayed in movies and television. But rarely do we get to hear about the science behind these DNA tests. Let's start with the basics. DNA contains the genetic blueprint for every human being. DNA stands for deoxyribonucleic acid. It is a molecule made up of repeating segments called nucleotides. Each nucleotide is made up of a phosphate group, a sugar group, and a base. The four possible bases are thymine, adenine, guanine, and cytosine. The bases are found in a variety of sequences. These sequences make up the code that provides your body with instructions on how to develop. The entirety of an individual's DNA is called its genome, and every person has a unique genome, with the exception of identical twins. Half of your genome comes from your mother, and half of your genome comes from your father. These basic principles give you enough background knowledge to understand DNA testing. Now let's take a look at some hypothetical examples. A sample of DNA can be obtained from a number of sources, including blood, saliva, or hair left at the scene of a crime. Forensic scientists only need a small amount of DNA because they can make more copies with a technique known as PCR. Then scientists can cut long strands of DNA into smaller pieces using restriction enzymes. The DNA is then put inside a gel that has a consistency similar to that of Jell-O. Then the gel is subjected to a voltage, which will cause the bands of DNA to run through the gel. Different DNA bands move through the gel at different rates. Large DNA strands move slowly, and short DNA strands move quickly. This results in a distinct pattern of bands in the end. That pattern of bands is what gives us useful information. Here you see bands of DNA that was found at the crime scene, along with bands of the DNA of 4 different suspects. As you can see, the bands from the DNA found at the crime scene match the bands from the DNA of suspect 3. That was an example of how DNA testing could connect a suspect with a crime. Now let's look at how DNA testing could be used for paternity tests. In this example, DNA samples were collected from a mother, 2 potential fathers, and a child. The strands of DNA were cut into smaller segments, just as in the previous example, through the use of restriction enzymes. After the DNA was subject to a voltage and run on a gel, we observe these patterns. The mother has a common band with the child. Bob has one band in common with the child and another band not shared by the child. All 3 of Larry's bands are shared by the child. This evidence suggests that Larry is the father of the child. Now, you hopefully understand some of the science behind DNA testing.